This recording is part 3 of 4 for Chapter 3, the cellular level of organization. We've already discussed that DNA is a very large molecule or macromolecule that holds all the genetic information for all living beings. This is where information that determines organisms' characteristics is found, and this information is passed down through generations. We know that the DNA is contained in the nucleus of the cell in the form of chromatin, and each cell has its own DNA. The DNA is a right-handed double helix, as you can see in this picture. DNA consists of long chains referred to sometimes as polymers, the term which means many units. These polymers are made up of single units or monomers that are called nucleotides. Each nucleotide contains a nitrogenous base, a sugar, and a phosphate group. There are 4 nitrogenous bases associated with deoxyribonucleic acid or DNA, including adenine, guanine, cytosine, and thymine. Nucleotides are joined to one another by way of covalent bonds between the sugar of one nucleotide and the phosphate group of another that make what appears to be the side rails of a ladder. As we look more closely at the structure of DNA, we can see the sugar-phosphate backbone of one of the 2 strands of the double helix, colored in yellow and blue, the yellow representing the phosphate portion and the blue representing the sugar portion. The nitrogenous bases, as you can see, are held together by way of hydrogen bonds. We remember that hydrogen bonds are the weakest bonds and are always identified by dashes instead of by solid lines. We remember that the 4 nitrogenous bases of deoxyribonucleic acid are adenine, thymine, cytosine, and guanine. Those nitrogenous bases occur in different orders along the strands of the DNA. However, you'll notice that they always pair with their complementary base pair despite the order in which they are found on one side of the strand. Let's take a closer look at that next. Here we have a picture of the DNA that's been untwisted, so we can see the sugar phosphate backbone on one side as well as on the other side of the molecule. You'll notice that the nitrogenous bases are held together by means of hydrogen bonds in the middle. No matter the sequence of nitrogenous bases on one side of the strand, the opposite strand must have a complementary nitrogenous base that will match the one on the opposite side. In other words, guanine and cytosine will always bond together. They always bond together because guanine and cytosine have places for 3 hydrogen bonds to occur between the 2 of those molecules. Adenine and thymine will always bond together. Those are complementary nitrogenous base pairs because they have places for 2 hydrogen bonds to form between the 2 of them. So no matter what the sequence on one side, the opposite side must have the complementary base pair in accordance with the nitrogenous base on the opposite side in order for a bond to form. Remember, DNA has 4 nitrogenous bases, adenine and thymine. They're often stated together because they will only bond with each other, whether thymine is on one side and adenine on the other, or vice versa. Here we have adenine on this side and thymine on this side. It doesn't matter. And cytosine and guanine will always bond together because they are complementary nitrogenous base pairs. Again, it doesn't matter which side it's on, but guanine and cytosine will always bond together. Understand that these nitrogenous base pairs will occur in different sequences, but their complementary bases must match. When we look at the contents of the nucleus, we know that the DNA exists normally in the form of chromatin unless the cell is going through a process of cell division. Chromatin is not just made up of DNA. In fact, only about 30% of chromatin is DNA. 60%, the majority of chromatin, is made up of special proteins called histones. These histones resemble little discs. The DNA double helix will wrap around 8 histone proteins, and it will wrap around twice to form a structure called a nucleosome. Again, a nucleosome is 8 histone proteins wrapped twice by the DNA double helix. Normally, chromatin, again, is only going to be seen in non-dividing cells, and in order for cells to go through cellular division, this chromatin has to organize itself. I often tell students that chromatin looks like cooked spaghetti. If a person reached into a pot of cold cooked spaghetti, it's all tangled up. It's very difficult to organize it and separate it without breaking it or it becoming tangled. So in order for the DNA to be divided after it's been duplicated, when cells go through cellular division, the chromatin has to condense into chromosomes in order for the DNA to be better organized. Just think of dividing uncooked spaghetti where it's all nice and condensed and easy to split apart. That would be a much more helpful process to divide something that's very well organized than something that's all tangled. Here we have a really good slide because it shows that cooked spaghetti appearance of chromatin as it normally exists within the nucleus of a cell. Again, this is found in cells that are not going through the process of cell division. When a cell starts preparing itself to divide, that means that the chromatin is going to start a process of condensing into chromosomes that are attached at a central location that is referred to as the centromere. The chromosomes consist of 2 sister chromatids. This is one sister chromatid on the left. This is the other sister chromatid on the right. They are joined together at the centromere, and within the centromere on each sister chromatid is a special molecule known as the kinetochore. When it's time to move the sister chromatids apart so that each new cell has its own copy of the original DNA, the spindle apparatus will actually attach to the kinetochore to direct movement. Kinesis means movement. At the ends of the sister chromatids are special structures known as telomeres. Telomeres are very similar to the plastic capping on the ends of shoelaces. Not that they're plastic, that's more of an analogy. The telomeres of the sister chromatids on the chromosomes are going to shorten each time the cell undergoes division. And that forms a clock so that the cells can only divide so many times. The telomeres shorten every time cell division occurs, so cells only have a certain number of times before they stop dividing because the telomeres will no longer allow them to divide if they have become too short. So again, they're like the protective capping on the ends of the sister chromatids. When we look at the picture of the chromatin, again, the chromatin wraps around these 8 globular histone proteins, forming a unit called the nucleosome, as those 8 histone proteins are double wrapped by the double helix of the DNA. In human cells, we have 23 pairs of chromosomes in our somatic cells. Remember, somatic cells are cells of the body that have all of the DNA from both of our parents. They are diploid cells. Somatic cells differ from gametes, which are sex cells, because gametes are haploid. They only have 23 chromosomes as opposed to 23 pairs. That allows an oocyte and a spermatocyte to join together to form a single organism after those stem cells divide, and differentiate. The DNA is important in our cells because stored on the DNA is our genetic code for how our bodies will be able to produce proteins based on the sequence of nitrogenous bases in the DNA that will be converted into sequences of amino acids to make a protein. Proteins are chains of amino acids that are joined together by dehydration synthesis to make a peptide bond between each amino acid. So if we have chains of amino acids, we have a polypeptide chain that creates a protein, in fact, the primary structure of a protein. As you'll remember, the primary structure of a protein is the chain of amino acids, whereas the secondary structure of a protein is when those amino acids start to form a beta-pleated sheet or alpha helix coil, depending on the attractions that those molecules have for one another, that will help them change configuration. A tertiary structure of a protein is when it further folds into a globular unit, and a quaternary structure is when 2 or more of those tertiary structures are going to add to one another through chemical attraction that holds them bonded together, and they form a functional protein. So again, the information stored on the DNA is going to be the sequence of nitrogenous bases. And every time we say these bases, we tend to say them in order, but they're going to be mixed up along that sugar phosphate backbone in order to indicate different types of amino acids. Every time we look at a triplet of nitrogenous bases, that will encode for 1 amino acid. If we look at this one strand of the DNA template, once the DNA has been separated, we see adenine, cytosine, cytosine. Those 3 nitrogenous bases are referred to as a triplet. The triplet will encode for 1 amino acid. A gene is a segment of the DNA that contains all the triplets needed and required to make a specific protein. Because DNA has the information on how to produce proteins, it has direct control over the synthesis of structural proteins, including things like the cytoskeleton, integral proteins embedded in the membrane, as well as secretory products. And DNA has indirect control over cellular metabolism by regulating enzyme production. Remember, all enzymes are proteins, and some of them may have a cofactor or coenzyme attached to them to help them perform their function. So we have this continual communication system between the cytoplasm and the nucleus, and the cytoplasm and the extracellular, which is crucial for the function of our cells as they communicate with one another in the form of tissues and then as they come together as organs. This particular slide, I'm not quite sure how it got in here. I think sometimes these things sneak in. This slide has to do with DNA replication as opposed to protein synthesis. So we're going to skip over this now, and I promise I'll make sure that it's in the next video that we go over so you understand the process of DNA replication. It's actually easier than protein synthesis. We're hitting the hard stuff first. So we know that ribosomes are the site of protein synthesis, and we know that DNA has the set of instructions on how to make a protein. DNA, however, is located in the nucleus and is too large to go through nuclear pores to get to the ribosomes that are either attached to rough endoplasmic reticulum or are free-floating in the cytoplasm. So in order to get the message off of the DNA, we have to produce a molecule that can exit out of the nucleus and take that message to the ribosome. That occurs in the form of RNA, specifically messenger RNA. RNA actually comes in 3 flavors. We have messenger RNA, which carries the written record or transcript of the instructions from the DNA out of the nucleus into the cytoplasm. We have ribosomal RNA, which forms ribosomes. Ribosomes are made up of large subunits and small subunits. And finally, we have transfer RNA, which is going to bring the individual amino acids to the ribosome so that they can be made into that polypeptide chain of amino acids, our growing and developing protein. If we look closely at this slide, you can see on the right-hand side those 4 nitrogenous bases involved in DNA. We have thymine, adenine, guanine, and cytosine. RNA is similar. However, we have one major difference in and of the fact that instead of thymine, RNA has uracil. RNA contains cytosine, adenine, guanine. However, there is no thymine. Instead, we have uracil. Pay close attention to that. The process of protein synthesis is, again, being able to produce a polypeptide chain of amino acids. If we look at the top picture, we can see an amino acid on the right being joined to this growing chain of amino acids joined together by way of peptide bonds. So remembering that that gene is just a portion of the DNA that's going to have the instructions on how to build one polypeptide chain, understand we've got over 25,000 genes that can code for proteins. Looking at the triplets on one of the 2 strands of the double helix of the DNA is where we have those triplet nitrogenous bases that will code for a specific amino acid. If we have adenine, adenine, adenine in a row on that template strand of the DNA, that forms a code for phenylalanine. If we have cytosine, cytosine, guanine on the template strand of the DNA, that can code for glycine. Why do we need to make so many proteins? We know that proteins are large, complex molecules. They play critical roles in the body. They do most of the work in our cells, and they're required not only for structure for the cell, but they also are so important in the function of the cells and regulation of our body's tissues and organs. Proteins are made up of hundreds or thousands of amino acids attached to one another in long chains. We call those polypeptide chains because the bonds that hold the amino acids together are peptide bonds. There are 20 different types of amino acids that the human body has that can be combined to make a protein. In an earlier slide, I showed you the 20 different amino acids. They're broken down into groups. We have essential and non-essential amino acids. Essential amino acids are ones that we have to eat. The non-essential amino acids are ones that our body can actually produce. All in all, we have 20 that can be combined to make protein. And it's the sequence of the amino acids in that primary chain of amino acids that's going to determine the unique three-dimensional structure and shape of that protein. And therefore, that determines its function, because function is always dependent upon structure. There's a bunch of different enzymes listed on the left of this slide, but just to show you, some of these proteins are involved in our immune system. There are complement proteins that enhance immunity, antibodies like immunoglobulins that again have a role in our immune system. Hormones, there are some proteins that act as hormones like insulin and growth hormone. Those are so important for our body's function. We also have proteins that act to transport lipids through our body. So our lipoproteins are important for that. And proteins can also act as buffers to help our bodies maintain a very specific pH. There are two main stages that are required to make proteins. The first one is writing down the instructions from one of the two strands of DNA after it's unwound. And that writing down of the instructions or getting the recipe is what we refer to as transcription. To transcribe means to write. This occurs in the nucleus. The second step is when the instructions that have been written down on the RNA strand are going to go through the nuclear pore, where they will go to a ribosome and form a protein. So transcription, or the writing down of the instructions, occurs in the nucleus. Translation, where those instructions are literally translated into a protein, is going to occur in the cytoplasm, specifically at a ribosome. The process of transcription not only involves writing down the instructions off of the DNA, the first RNA molecule that will be formed is actually a molecule known as pre-messenger RNA. Pre-messenger RNA will be converted into messenger RNA, and it is technically the messenger RNA that will exit through the nuclear pore to be transported to a ribosome, where it will be translated at the ribosome to form that polypeptide chain of amino acids, which is our developing protein. When we look at transcription, when the DNA is unraveled, and we'll talk about that in just a second, we'll notice that there are two strands that are exposed from the DNA double helix. One strand is referred to as the template strand. The template strand is used for this developing molecule of pre-messenger RNA. Remember, RNA does not contain thymine. So if we look at the base pairs that are being joined to the exposed DNA template, with the help of the enzyme RNA polymerase, the appropriate complementary nitrogenous base pair is going to be written in sequence in accordance with what is on the DNA template strand. Where we have adenine on the DNA, since we don't have thymine in RNA, a molecule of uracil will be placed. We do have adenine to go with the thymine on the DNA, so the next molecule added to the developing RNA will be adenine. Here we have guanine next, so with guanine we're going to match it with cytosine on the RNA. Here we have adenine again on the DNA. We don't have thymine in RNA, so we have to use uracil. And that's how we develop our growing pre-messenger RNA molecule. We're still in the nucleus. In the previous slide, we saw that there is a molecule of pre-messenger RNA that's produced as RNA polymerase starts to build that molecule of pre-messenger RNA. On the DNA are long regions that are non-coding regions for amino acids. Scientists are not quite sure what their function is at this point, so in order for that pre-messenger RNA molecule to have those regions taken out requires a spliceosome that will help to cut out certain portions of the pre-messenger RNA molecule and glue together the remaining portions that are going to act as the final messenger RNA. In this process of splicing by the spliceosome, portions of the pre-messenger RNA molecule that are cut out are referred to as introns. Introns stay in the nucleus. They don't exit the nucleus to become part of the protein as it's built in the cytoplasm by the ribosome. The remaining portions of the pre-messenger RNA molecule are referred to as exons. Exons are spliced together, and as they're joined together, they carry the coding sequence for the polypeptide chain of amino acids as they form that final messenger RNA molecule, which will travel out through the nuclear pore to hook up with a ribosome to start making a protein. When coding regions are spliced out, different variations of proteins are made, giving a much larger variety of possible proteins and functions. Remember, introns stay in the nucleus. They're not part of the messenger RNA. Exons exit the nucleus as the messenger RNA molecule. The process of transcription is going to begin when special gene activating chemicals that are referred to as transcription factors stimulate the histone proteins to loosen at the site and help position RNA polymerase, which is going to direct that production of pre-messenger RNA. The transcription factor will bind to a special portion of the DNA sequence, which is referred to as the promoter section of DNA, and that's the beginning of the gene on the DNA where transcription begins. Transcription occurs on the template strand. There are two strands that are exposed when the DNA double helix is unwound. There is a template strand, which in this picture above my head on the video, the bottom part is the template strand because that's the part of the strand being used as a template to make pre-messenger RNA. The opposite strand that's exposed is referred to as the coding strand because the code on the coding strand is similar, not exact, but similar to the sequence of nucleotides as the pre-messenger RNA molecule being built below. Remember, it can't be exact because RNA has uracil, whereas DNA has thymine instead of uracil. There are 3 stages of transcription. Initiation is the initial stage where RNA polymerase, with the help of transcription factors, will bind to the promoter area on the DNA double helix. The RNA polymerase will help that DNA double helix to unwind. The next stage of transcription includes elongation, where incoming nucleotides, RNA nucleotides, with the help of RNA polymerase will be aligned with the complementary DNA nitrogenous bases on the template strand, and the RNA polymerase will link those together. If we look at the middle picture, we can see the template strand of DNA on the bottom, and where we have adenine, the pre-messenger RNA molecule should have uracil and shows guanine. Oh dear. Okay, well that's a mistake. Where thymine is on the DNA, we have adenine. Where adenine is on the DNA, we have uracil. That's correct. They goofed on the picture. Where guanine is on the DNA, we have cytosine. Here's guanine, cytosine, thymine, adenine, thymine, adenine, adenine, uracil, adenine, uracil. So they goofed initially. Oh well, that's going to be a genetic defect. Finally, we have termination. Termination occurs when the RNA polymerase reaches the terminator sequence or termination sequence on the DNA. And I'm going to take my little picture off of here for a second. That ends transcription, and the newly formed pre-messenger RNA will separate from the DNA. Remember, a spliceosome will go through the pre-messenger RNA and cut out introns, which will remain inside the nucleus. The next part of building a protein is translation. Translation is literally taking the language of a sequence of nucleic acids and translating it into a sequence of amino acids. For each triplet on the DNA, the corresponding triplet on the final messenger RNA is referred to as a codon. There are 64 codons, 3 of which are referred to as stop codons because they stop the process of producing a protein, one of which is called a start codon and can also code for the amino acid methionine. In order for the amino acids to get to the ribosomes, there is yet another form of RNA required called transfer RNA. Transfer RNA has anticodons with complementary base pairs that match the codons on the messenger RNA. In this picture, we can see the ribosome comprised of the small subunit and the large subunit above. We can see the messenger RNA with its triplet codons, and we see the anticodons attached to the transfer RNA molecule. Let's look at the very middle codon right here in the center. We'll notice that the codon on the messenger RNA is cytosine, guanine, uracil. The anticodon on the transfer RNA molecule has to have the complementary pairs for cytosine, guanine, and uracil. So the anticodon that you'll notice on the transfer RNA molecule in the center has guanine, cytosine, adenine, which are the complementary nucleotides for the codon, which has cytosine, guanine, and uracil. That's how these individual amino acids are brought into the proper sequence for the growing protein. Now, when amino acids are attached to the transfer RNA, they become attached to transfer RNA with the help of a special enzyme called aminoacyl transfer RNA synthetase that helps put the amino acids on there in the first place. And again, that particular amino acid must match the anticodon on the transfer RNA. This particular slide pretty much shows everything, so I'll talk you through it. I'm going to take my picture off. If we look at the upper left portion of this slide, you can see that within the nucleus, the DNA has been unraveled, and the template strand of DNA, with the help of RNA polymerase, is producing pre-messenger RNA. Through the process of splicing with the help of a spliceosome, the pre-messenger RNA molecule is going to turn into the final messenger RNA molecule because the introns have been cut out and only the exons are part of the messenger RNA molecule, which goes through a nuclear pore into the cytoplasm. So here we have our freshly made messenger RNA molecule coming out of the nucleus, traveling to a free ribosome or an attached ribosome to be translated by a ribosome into a protein. With the process of translation, that also includes initiation, which occurs when four components combine. We have the small subunit of the ribosome combining with the large subunit of the ribosome, and an initiator transfer RNA molecule that carries the amino acid methionine. Methionine is the initial start code for a building growing protein. We always start with methionine. Then the messenger RNA molecule will attach once the transfer RNA molecule has attached in its site. And the large ribosomal subunit, once all of this has happened, is going to take the mRNA molecule through the process of elongation. As the messenger RNA molecule passes through the ribosome, the process of elongation means that we're adding amino acids one at a time to this growing polypeptide chain that has 3 repeating steps. First, there's codon recognition, where the codon on the messenger RNA is recognized and matched with the anticodon of an incoming transfer RNA molecule that's going to combine with the codon on the messenger RNA. This occurs in this particular site of the ribosome, the A site. So we have incoming amino acids on the A site. We started with our initial amino acid on the P site. The 2 amino acids between the P site and the A site will form a peptide bond by the process of dehydration synthesis. And once that peptide bond is formed, you'll notice that the empty transfer RNA molecule at the P site is going to be shifted over to the E site. So the new peptide, the new polypeptide, goes over or bonds to the right, and then the empty transfer RNA molecule is going to shift over to the E site on the large ribosomal subunit, and it will then be released. And that's what we refer to as translocation. So the entire ribosome is moving along the messenger RNA, and it locates things in different spots. That's what translocation refers to as it moves along the messenger RNA. New amino acids are going to be brought by transfer RNA to the A site. They will form a peptide bond, and then the transfer RNA molecule in the P site will shift over one to the E site, where it eventually gets released. After this empty transfer RNA molecule has been released, remember that the amino acid enzyme called amino acetyl synthetase will join that transfer RNA molecule with the proper amino acid yet once again, so it can be used again. Finally, when a stop codon on the messenger RNA molecule is reached, there are 3 different stop codons: uracil guanine adenine, uracil adenine adenine, or uracil adenine guanine. When one of those 3 stop codons is reached on the messenger RNA molecule, that signals the end of the growing protein, and that causes a release factor to bind to the A site of the large ribosomal subunit, so no new amino acids can be brought in. When proteins are produced within the rough endoplasmic reticulum, what happens initially is there is a special particle called the signal recognition particle that, along with an endoplasmic reticulum signal sequence, will bind to the messenger RNA ribosome complex. They'll bind to that complex, and then the signal recognition particle will help that complex bind to a receptor site on the rough endoplasmic reticulum. Once they're bound to the rough endoplasmic reticulum, the signal recognition particle will be released. So this little black ball here, or gray, dark gray, will be released, and the growing polypeptide is going to come through the pore of the rough endoplasmic reticulum into the cistern. When that starts occurring, the green signal sequence will be clipped off by an enzyme embedded in the membrane of the rough endoplasmic reticulum. As protein synthesis continues, as this polypeptide chain continues to grow, sugar groups may be added, as is demonstrated in this case, by proteins enzymes embedded in the membrane of the rough endoplasmic reticulum. That will modify this growing protein, and we end up with a completed protein being released from the ribosome, folding into a 3-dimensional conformation, the process of which is aided by molecular chaperones, which will also direct it toward the end portion of the rough endoplasmic reticulum in order for part of that membrane to pinch off, forming a protein-coated transport vesicle that will carry this glycoprotein to a Golgi apparatus for even further modification. RNA codons represent amino acids. There are 20 different amino acids, but there are different combinations of codons that might represent amino acids. For instance, if we look at the chart on the left, you'll see that there are 6 different codons that can encode for the amino acid leucine. Here we have 6 different codons for here and 2 here for the amino acid arginine. Whenever we see the codon adenine, uracil, guanine, that is a specific codon for either the amino acid methionine, or it can be used as a start codon. And there are 3 different codons that will stop the process of protein synthesis, including uracil adenine adenine, uracil adenine guanine, and uracil guanine adenine. In going from DNA to protein, we have to remember that our DNA has genes. Genes are instructions on how to build a protein. When the double helix is unwound, instructions are taken off the DNA template strand, which contains nitrogenous bases that are read in triplet. If we look at triplets of nitrogenous bases on the DNA template strand, we see things such as this first triplet of thymine, adenine, and cytosine. When that is transcribed or written into messenger RNA, the messenger RNA contains complementary base pairs of the nitrogenous bases from the DNA. So the complementary nitrogenous base pair for thymine in the first triplet is going to be adenine in the codon of messenger RNA. The second nitrogenous base in this first triplet, being adenine, is going to be matched with its complementary nitrogenous base pair as uracil, because RNA does not contain thymine. We know that cytosine, the last nitrogenous base in the first triplet, is going to be paired with guanine in the messenger RNA. When messenger RNA is translated by matching anticodons from transfer RNA molecules to the codons on the messenger RNA to bring those specific amino acids into their specific place in this growing chain of amino acids, the polypeptide chain, we can notice that the anticodons are going to look exactly like the original triplet in the DNA template, with the exception of the substitution of uracil for thymine, because RNA does not have thymine. If we look at the first anticodon brought in by the transfer RNA molecule, we see uracil. Well, the triplet in the DNA template stand, since we don't have thymine in RNA, the thymine is going to be replaced by uracil in the transfer RNA. But notice the other 2 nitrogenous base pairs in that first triplet are exactly the same. So what happens is the anticodons from the transfer RNA molecules will always match the original DNA template strand, with the exception of the fact that wherever thymine exists on the original DNA template strand, we will substitute it for uracil in the anticodons for transfer RNA. let's look at triplet number 3, because there's no thymine in that triplet. We have adenine, guanine, cytosine. By the time we go through transcription and get back to translation, you'll see that we're translating it into the exact same thing. Wherever we have thymine on the original template strand, we're going to see it substituted for uracil, but everything else is exactly the same. And I know I'm not using triplets per se, I'm just going by the strand and showing you the difference. However, things are read in triplets, converted to codons and anticodons. I just wanted to prove the point that the anticodons that are making this polypeptide are actually being translated back to the original code that was in the DNA, with the exception of where there was thymine in DNA, we have uracil in the RNA. I hope that explains that well for you. This ends this recording. I'm going to go on to part 4.